Okay, so we are going to discuss, let me give my video a label here. And let's also note that it's also called loop of Henle. That's the person who figured it out. So what we're going to do is just describe very broadly, before we even get to the Nephron loop, I'm going to do some very basic biology. And that is because the whole way the Nephron loop works is really based on osmosis and using permeability to sort of hack osmosis in a way that's working for water reabsorption and working for the kidney. So I'm going to make a box. And the reason I'm making a box is because I just want to isolate my environment. So I want to be able to control what's happening in the box. And in my box, I have 2 tubes. Make them a little bit different colors just so I'm too much orange. Let's have a pink one. There we go. I want to make my red tube a little bit larger. No, I want red. And I want circle. There we go. Sorry, I'm just trying to make my tubes large enough for me to draw in. I'm having a really hard time doing that, apparently. There we go. Okay, so I have a controlled environment. I have 2 tubes. Let me make a little key here. So blue dots are water, green dots are sodium chloride, aka salt. So in each box, I have a bunch of water. And the relative amount doesn't matter right now, so don't try and look for meaning in the difference between the number of dots. There's not one. So this is my starting condition. I have 2 tubes. The tubes are next to each other, and they both have water and sodium chloride in them. Now, adding to that mix, I'm going to introduce a pump. Now, pump implies active transport, meaning I'm spending ATP to move something. And in this case, what I'm moving is sodium chloride out. So I'm writing it out here, but I'm going to just abbreviate it as dots. So the pumps are going to be constantly turning over and spending ATP out. And because the pumps are always going as long as they have enough ATP, and I have my, box which is a controlled environment, as long as the pumps are active, we're going to see sodium chloride build up in the interstitial space. So that ends up giving us a condition, and don't worry, I'm going to redraw my water dots here. Giving us an internal condition where the red tube, because it's moving sodium chloride out, has more water in it and less sodium chloride. Now let's add our permeabilities in. So pink tube is only permeable to water. It's not permeable to sodium chloride. And the red tube is permeable to sodium chloride due to the pump. Is impermeable to water. So that's why the water stays in. It can't get out. It can't leave with the sodium chloride. And it is permeable to sodium chloride. So if I know that I'm always pumping sodium chloride out of the red tube, meaning that I'm consistently keeping the area around the tubes high in sodium chloride, take a moment to think about what that means, what's going to happen next in this picture, looking at the pink tube. And remember, we're recording, so you don't have to use your voice if you don't want to, but take a moment to think about it. And then I'll tell you. Okay.
Could you repeat the question? Sorry.
Yes. So if I know that I'm having a sodium pump, pumping sodium chloride into the interstitial space here, and it's always going because I'm always giving it ATP to spend. And I've told you about these permeabilities down here. What is the next thing that's going to happen in the picture?
Isn't it like water follows stuff?
Yes, exactly. So the next thing to happen, if I tell you that the pink tube is water permeable, and you see this environment on the outside of the tube where there's lots of sodium chloride, osmotically, water is going to follow that sodium chloride. So you're going to have the passive movement of water out of the tube, not because of active transport directly, but because the outside of the tube is
higher concentration
of salt, lower concentration of water.
Yes.
So you can also summarize that this domain is active transport, and this domain is passive transport. But the passive transport over here is only happening because of the active transport of sodium chloride over here. So when the book talks about sodium chloride pump-driven osmosis, what it means is the osmosis that's happening in the pink tube is only happening because of the action of its neighbor. If the sodium chloride pumps were to stop transporting the sodium chloride, then you would see osmosis slow down and eventually cease as the system moved toward equilibrium. So this is my very basic starting condition. You can see that pumping of salt into the interstitial space drives water out of the pink tube and into the interstitial space as well. So up until now, I've just sort of shown you this controlled, rarefied environment. Now let's apply it to the loop. Because let's say the circles were not tubes, they were spheres instead. Eventually, the sodium chloride pump would run out of sodium and chloride to pump, right? If there were just, a cell. And that would mean that eventually osmosis would stop and the system would equilibrate, and then you'd get no further transport. And that's because you have a closed system. There's no input, there's no output. The nephron loop, of course, is a system with input and output. So let's draw that. And I know that the book stresses the thick and thin aspect of the tubes. That's just referring to their histological appearance. The thickness and thinness stuff is not important for understanding how this transport works. So I'm not going to draw the thick and thin business at all. I'm just going to stick with my color scheme that I had initially. And we're also going to acknowledge that there's flow of filtrate So instead of having a closed system where there's fluid that's stagnant inside of two neighboring tubes or bubbles, now I have bulk flow of fluid moving through the tube like water through a pipe. So let's draw our transport pumps here. And I'm just drawing my sodium chloride, so we know that these pumps are spending ATP to move that sodium chloride out into the interstitial space. So what we would see is as we have filtrate entering the tube, we know that this pink area is water permeable. So at the start, there's going to be a lot of water in this tube, but it's going to move out towards the sodium chloride in the interstitial space. And so you see less and less of it as you move down the tube. Now there's also sodium chloride in the filtrate at this point. So what you see as you move down the descending limb is that you go from having about equal parts water and sodium chloride to having mostly sodium chloride. And then it's that sodium chloride, so you see a decrease in sodium chloride as you move up the ascending limb, but your concentration of water compared with the sodium chloride is higher. So you might be asking, well, if water is moving into the interstitial space, why doesn't the system just equilibrate because now there's equal amounts of water and sodium chloride? And the answer is because the water doesn't stay in the interstitial space. There is a specialized capillary bed that comes off of the efferent arteriole called the vasa recta. And that is referring to the fact that it's a little capillary bed that looks like a ladder. Oops, I wanted blue. There. and it looks like a ladder because it's wrapping around the descending limb. So when the water diffuses out of the descending limb due to osmosis, it doesn't stay in the interstitial space with the sodium chloride. It goes into the blood via the capillaries of the vasa recta, and then that water from the filtrate that you've just extracted just goes right back into your venous blood. So it's a way to make urine while not losing water. So let me just draw the rest of this real quick. So we've got efferent arteriole. So let me think about how I want to draw this. There's my glomerulus. There's the afferent. I'm going to label all these. And this laddery structure is the vasa recta. And then if I continue, so there's proximal convoluted tubules right there. So the reason that the yellow arrows are important is because, of course, as you're filtering filtrate out due to glomerular filtration, so remember there's that net filtration pressure difference that keeps filtrate being formed, you're always having flow of filtrate through the tube, which means that there's always fresh filtrate coming down, so that you're always taking water out of it because you're always having filtrate come up this way and pump sodium chloride out. So due to that, as long as you are making filtrate, the loop reclaims water from that filtrate. And the reason that we refer to the water reclamation here in the loop as driven by the sodium chloride pump is because the pumping action, moving sodium chloride into the interstitial space, is what causes this osmosis. If that didn't happen, if the pumping didn't happen, the osmosis would also stop. And then the reason the system does not reach equilibrium is because the direction of flow in the tubes is opposite and the tubes are right next to each other. So the fact that the tube flow is opposite and the tubes are positioned next to each other is why we call it the countercurrent. So countercurrent referring to the opposite flow directions, multiplier, and that's just referring to the fact that it multiplicatively enhances how much water is being taken back from the filtrate. So there's a lot of pictures and a lot of terminology, but if you kind of break it down to what you know about osmosis and permeability, it makes a lot of sense. And the final thing I'll say about this is that this is obligate water reabsorption. And obligate refers to the fact that the loop works the same regardless of how hydrated or not hydrated you are. So you can and absolutely do adjust your urine concentration or osmolarity according to how hydrated you are. So if you're dehydrated, you're going to be making very small amounts of very concentrated urine. If you're very well hydrated, you'll be doing the opposite. But that's not contingent upon the actions of the nephron loop. The nephron loop works the same way all the time, regardless of your hydration state. And that is the nephron loop and countercurrent multiplier.